So, to start, could you give us a brief history of who you are and the key goals you're aiming to achieve in your work?
My name is Beth Shapiro. I am currently the Chief Science Officer at a company called Colossal Biosciences. I'm also a professor in the Department of Ecology and Evolutionary Biology at UC Santa Cruz, where I run a lab called the UCSC Paleogenomics Lab. I've worked for a long time in a field called ancient DNA, where we go out into the field and we collect remains of plants and animals that used to be alive, and we extract DNA from those remains to try to reconstruct ancient ecosystems and learn things like why did some species become extinct when others survived, or what makes some communities or ecosystems more resilient than others to rapid changes or perturbations to their habitats. All of that work pushed me into a place where a lot of people asked me a lot of times what it meant about bringing this DNA from the past back to life and whether this could be translated into bringing actual extinct species back to life. So in 2015, I wrote my first book. It's called How to Clone a Mammoth. It was really a long form answer to that question that said, it's going to be really hard to do something like that. And then, I got more involved in thinking about the technologies and not just what those technologies might mean for this idea of bringing extinct species back to life or de-extinction, but also how those technologies could be used to stop living species from becoming extinct. And with all of that in mind, I transitioned a couple years ago to my job at Colossal, where we are a de-extinction company and a species preservation company. We're developing those tools necessary to bring extinct species back to life and also working with nonprofit and conservation organizations around the world to use those tools to stop species that are currently in danger of becoming extinct from following that same path.
Mhm. So you've gone from Allentown, Pennsylvania to Rhodes Scholar at Oxford to professor at UC Santa Cruz and now Chief Science Officer at Colossal Biosciences, which you outlined. Looking back, which transition felt the most improbable to you at the time and why?
Well, that's an interesting question. I think every transition feels improbable. I've really based my life on making decisions based on something that I'm excited about, by keeping my mind open, by being open to trying new things or taking risks when necessary. I think because it's most proximate to where I am now, the transition from professor at UC Santa Cruz to chief science officer at a company is probably the most improbable of these, although I probably would have had a different answer if you asked me at all the different stages of my life.
Mhm. So how has becoming a parent changed the way you think about risk, long timelines, and the future you're working towards?
These are very long and intensive questions here. How has becoming a parent changed the way I think about risk, long timelines, and what was the third thing?
And the future you're working toward, just more generally.
The future I'm working toward. How has it changed my mind about risk? Well, obviously, if I think about risks, when you are a parent and you have to care for another person and make sure that they are happy and healthy, you have to consider your own personal risks in a different way. I do think that I consider um, maybe thinking of it in a different way than most people do. I think when people think about the biodiversity crisis that we're going through right now as a global society, we often think of doing something, interfering with the natural way or manipulating species or manipulating ecosystems as taking a risk, whereas the other thing, not doing any of that, is not at all risky. But I think that that is not at all true, that both of these are decisions and both of these carry inherent risks. And if we just continue along doing exactly what we're doing right now, we know what the consequences of those decisions are. And as a parent who I'm thinking about my kids, I'm thinking about their kids, maybe the future of the planet, I think that not allowing ourselves the opportunity to explore what biotechnologies might be able to do to help us save species, to help create a future that is both biodiverse and filled with people might be. I think we don't want to risk not doing that because of the way I'm thinking about risk. Your second question was, how has a parent influenced the way I think about long timelines. I'm not really sure how to answer this one. I mean, everything operates on a really long timeline. I'm an evolutionary biologist, and so I understand that life is essentially a really long timeline. And I guess I just think of the path that we're on is a very short part of the path of all of life on this planet. But we do have an opportunity to influence the way that that goes. I'm not sure that answers your question
Mhm.
about long timelines, but I'm also not really sure what you're going for there. And the
Mhm.
third part was risk, long timelines, and
The future you're working toward.
the future I'm working. Oh, I think I probably answered that one in
Yeah.
the first one as well.
Regarding risk, I didn't really realize this when I was asking the question, but earlier today I interviewed the head of risk at Gold or former head of risk at Goldman Sachs, and now all he does is climate policy work, and he's saying very much similar to what you're saying, where to not consider this huge issue is far more risky than to continue as we're going right now. So that leads me to, so what is something about working with ancient DNA that people almost always misunderstand, even well-educated science fans?
Everybody always thinks of Jurassic Park. That's the first thing they think about. And they imagine that we're going to be able to go out into the world and get a mammoth bone, and then we're going to stitch together the pieces of mammoth DNA that are there, and then we're going to end up with a mammoth. And that's how Colossal's working on things, or that's how other people thinking about de-extinction are working on things. That's not how we're working on things. Instead, when we go and we get a mammoth bone and we extract DNA, there's a ton of mammoth DNA in there, but it is all broken because as soon as an organism dies, the DNA in all of its cells starts getting chopped up into smaller and smaller pieces by things like UV radiation, which also hurts our DNA when we go outside. That's why we wear sunscreen. But we, of course, have proofreading enzymes that will come and repair the damage made by UV. But that's an energy requiring process. Once you're dead, there's no more energy. So the damage caused by UV just accumulates in the DNA. Also, freezing and thawing, the water will expand and physically shear the DNA molecule. That will damage DNA. And things like microbes, fungi, and bacteria that get into the bone or the mummy or whatever it is and chew up the DNA to transform it into the next generation. All of these processes start right away. So when I get a mammoth bone and it's 30,000 years old and I extract DNA, I have a ton of DNA. But like if I were to take a swab of my own cheek and sequence DNA, I could get strands of DNA that are hundreds of millions of letters long. In a mammoth bone, most of the DNA that I get is shorter than 60 letters long. So I have to piece them all together. But I do this using a computer and using a closely related species as a scaffold. So mammoths are most closely related to Asian elephants. That's the closest living species to a mammoth. In fact, mammoths and Asian elephants are more closely related to each other than Asian elephants are related to African elephants. So they're very closely related to each other, about 5 million years diverged, which means that an Asian elephant genome is already more than 99.5% identical to a mammoth genome. So I can take all those little tiny fragments of mammoth DNA and use my Asian elephant genome as a scaffold, and then use a computer to figure out where on this scaffold each of these tiny little pieces goes. And eventually I can build up enough of those little fragments that I can see the whole mammoth genome. So how would we clone a mammoth? Well, you can't clone an animal that's already dead, but with a living Asian elephant cell, I have something that's alive that I could clone using the same process that most famously brought us Dolly the sheep. But first I have to edit it. And the way I decide how to edit it is I look at all of my mammoth genomes and I compare them to all of the Asian elephant genomes. And I ask, where are the mammoths all the same as each other, but different from the Asian elephant? And those are the places in the DNA code that I want to then go into that Asian elephant cell and gradually cut out the elephant version and paste in its place the mammoth version. So I'm not stringing together bits of mammoth DNA that are old and ancient and using something else to fill in the holes. In Jurassic Park, they used frog DNA to fill in the holes for some reason that is confusing to me because even then we already knew that birds are dinosaurs, right? So I don't know why they used frogs, but whatever. It was a movie, not a documentary. We're not doing that. Instead, we starting with the elephant genome and we're gradually tweaking it using the tools of genome editing, genome engineering to transform it into a mammoth genome. And then we will have something that we can clone.
Hmm. So when you say 5 million years diverged, what exactly do you mean by that? When you
That
referring to
means
the
that they shared a common ancestor around 5 million years ago.
Hmm. So ancient DNA work can be incredibly failure prone with contamination, degradation, dead ends. How do you personally handle that level of repeated failure?
I think you just look good. You look forward to the good times. I mean, we do whatever we can to avoid contaminating our data. We have a specialized facility that we work in where we wear a full bodysuit. It has positive air pressure. We do UV radiation to the surfaces to break down any DNA that might be left there. We're constantly bleaching things. I don't make my students swim in bleach, but if I could, I would. No, I'm just kidding. I wouldn't do that. But anyway, all of these things stop our DNA from contaminating the DNA that's there. And then we just develop really cool tools, computational tools or lab tools that we can use to tell the difference between a contaminant and a real DNA sequence. And you can gain a lot of confidence in the data that you're generating based on using the types of tools that are available today.
Hmm. What's the most surprising story behind a sample you've ever worked on? Where it came from, how it was found, and what it revealed, or just
That's an interesting question. I've worked on a lot of weird samples. I think the one that is most interesting to me right now, and it's because the story isn't finished yet, is many years ago we were working in northern Canada, in the Yukon, where there's gold mining that's actively happening. And where there's gold mining, they're washing away the permafrost, the frozen dirt that contains these bones, to try to get to the gold-bearing gravels underneath. And as they're doing that, the bones are exposed that we can use to extract DNA. And a few years ago, we found a really beautiful skull from a horse from some permafrost that we know is older than 700,000 years. It's very rare to find samples that are that old. Most of what we find dates to within the last 100,000 years or so. So this is an exciting bit of land to be working on and a really beautiful fossil. And also, most things that are that old don't have any surviving DNA. So this beautiful fossil found in frozen dirt was a real opportunity to see if we could get super old DNA. So we got this horse DNA, we extracted it, we were very excited about it. There really was DNA. We got a ton of DNA. We were able to sequence and assemble a whole genome. In the meantime, we sent the teeth to some horse experts who looked at it and said, this is a sample that belongs to the same lineage of horse that was later domesticated in Asia and in Europe that we all know today as domestic horses, Equus caballus. So we extracted DNA, mapped it to that genome, anticipating the perfect result, and it's not a horse. It is some species that was unknown to paleontology that is more closely related to donkeys than it is to horse, but we didn't know that this species existed. We're calling it preliminarily a woolly donkey right now because, we have to name it and go through the whole process of describing the taxonomy and describing the DNA. We've since found other horses, horse-like animals that belong to this same species, so we know it's not just a one-off, but discovering something that is entirely unknown and unexpected, I think, is probably one of the coolest things that I've been able to do in the field so far.
you've worked extensively on reconstructing the genomes of extinct species like mammoths and dodos. What have been some of the most surprising discoveries from your ancient DNA studies?
Well, new species of horse is one. I think one of the things that has been surprising to me and fun to learn about is how often we see gene flow between closely related species. This is something that we don't really think of. We imagine that once things are different species, that means, because that's what a species is, that they don't reproduce. But we have to remember that people made up those classifications, and biology sometimes doesn't care. And the first time we learned about this, and it really struck everybody, was when it was discovered that humans, after dispersing out of Africa, bred with Neanderthals, and that all of us have a little bit of Neanderthal DNA in our genomes because of this admixture, we call it, or interbreeding between lineages that we would like to think of as different species. We now know that this was really common in evolutionary history. Brown bears and polar bears interbred every time they overlapped in range in the past during the ice ages when it was really warm. And brown bear habitat got pushed really far north and overlapped with polar bears. They interbred. When it's really cold and polar bear habitat gets pushed down really far south into brown bear habitat, they interbred. And all of these animals today have residual evidence of that interbreeding. We know that wolves and coyotes interbreed, that different species of elephants have interbred. Like the more we look at DNA, the more we find that this has been really common through evolutionary history. I think that's kind of fun.
In your role as Chief Science Officer at Colossal Biosciences, what progress has the company made toward de-extinction, and what challenges remain in bringing back species like the woolly mammoth?
Every species that is a candidate for de-extinction faces a different set of technical challenges, ecological challenges, ethical, social, etc. And therefore, we have a long list of things that we need to solve for each species. What I like about working at Colossal is it's an opportunity to write all of these things down and then try to solve them a little bit at a time. We've been able to do a lot of things recently. We published a paper with our woolly mice in it. I don't know if you saw that, where we were able to put together as many as 11 different DNA edits in the same animals and have these very adorable, cute little woolly mice running around. We were able to edit the genome sequence of a surrogate genome gray wolf at 20 different sites and birth the dire wolves that have a white coat, which we engineered by manipulating their DNA. And they're larger and they're more muscular than gray wolves. And these are all traits that we learned by sequencing DNA from dire wolves. We've recently made huge progress in our bird de-extinction project. One of the challenges of working with birds is that you can't clone birds using that process that brought us Dolly the sheep, where you take a cell and you turn that cell into a living animal. That's because you have to have an egg cell that is ripe, that you remove the nucleus from, and then you transfer the nucleus, that's where the DNA is, into that empty egg cell. That's how we got Dolly the sheep. In birds, we can't get those egg cells at that stage. When a chicken is egg is first laid, there's a day-old developing chicken there, and you cannot just remove one nucleus containing the DNA and then shove something else in its place. So we need an entirely different approach to editing bird DNA and have those edits passed on to the next generation. And we made a significant leap there a few weeks ago, where we were able to learn the recipe that we need to keep cells alive in the lab that will eventually become either sperm or eggs. They're called primordial germ cells. So these are the cells that we will edit in birds. Here's a cool, crazy story. The way that we will make a dodo is that we will edit sort of pre-sperm cells and pre-egg cells in the dish in the lab. And then we will make chickens, not pigeons, dodos were a type of pigeon, but chickens that can't make their own sperm cells or egg cells. And when those chicks are developing, we will inject our edited sperm or our edited egg cells into those developing chickens. And when those chickens grow up, they will be entirely normal. We can mate them together, and those entirely normal chickens will lay an egg that hatches into a dodo.
Mhm. Very cool. Do you think this is something I didn't write about, but just as hearing you, do you think AI will have a role in, I don't know, forgive me for my ignorance. I don't know very much about the field, except sort of like estimating what an animal's DNA was and helping you synthesize something like that, or not so
I'm
much?
not sure that AI will help us estimate what the DNA was. We have pretty sophisticated sort of algorithms that we use to piece together the broken bits of DNA. But where AI is going to be really helpful first, I think, is mapping the letters in our DNA to the way an animal or a plant looks or acts is one of the hardest problems in biology. We've sequenced a lot of genomes, but it's like having a whole bunch of encyclopedias that don't actually have the entries on them, or having a whole bunch of phone books, but nobody's name on it. Like we know a lot of information, but we can't actually carefully, in most cases, link that information to what it actually does, which means that if I want to take a a gray wolf and turn it white, right? I might not know how to do that. I might not know what genes to tweak. Now, it so happens that hair color is one of the things we do know a lot about, so we could do that in the case of our dire wolves. But if I want to make something bigger or, really bigger, we know that there are lots of different genes that are involved in size in people, right? We don't know exactly which ones they are. We know which part of the genome they're in, but AI will be able to take all of the information that we have generated for people, for fruit flies, for bison, for cattle, for everything that's out there, and pull it all together and make really creative hypotheses about what DNA we should start to modify. So I think that's going to be the where we see the first boon for de-extinction research, or really any sort of research. It's also valuable for thinking about genetic disease. How do I know what DNA sequence or what change it is that causes this particular disease? Well, I don't know, and there's a lot of soup in there, right? I got to figure this all out. But if an AI can suck in all of the human genetic information that's ever been produced, and there are millions of human genome sequences that are out there, with phenotypic data, with all the information that we have online from experiments that scientists around the world have done, an AI is much more quickly than a human could going to be able to narrow down the set of hypotheses about what's causing disease and give us a path toward creating a cure.
Hmm. So, when you wrote How to Clone a Mammoth, you argued that we will never clone a pure mammoth, only engineer elephants that carry mammoth traits. 8 to 10 years later, has anything in the science made you update that view?
No. When we talk about de-extinction, we're not talking about bringing back something that is identical in every way to something that used to live. And this is not only because right now we don't have the technology to synthesize whole chromosomes or get them into cells. Maybe eventually we'll have that technology. But even if I could create something that is 100% genetically identical to a particular mammoth that used to be alive, the environment that that animal is born into is different. And we know that environment influences the way that our genes behave. Everybody knows clones, right? Do you have any friends who are identical twins?
Yeah, yeah, sure.
They're clones. They're
Mhm.
genetic clones of each other. But they're not the same person, are they? You couldn't swap one of them out from another and imagine that it's the same person. The
Mhm.
same would be true in this case. Now, another question is whether we would want to. Why would I want to make, if I'm trying to create a population of animals that's capable of thriving in the habitats of the future, why would I want all of them to be identical to a particular individual that lived at some point in the past? If we're using an elephant as a base for them and they're going to live in places where elephants lived, those elephants will have evolved defenses against diseases that are circulating today that that mammoth from the past wouldn't have had any defense against. So I don't want to make it exactly like that mammoth. I want it to be able to thrive in the habitats of the future. So when we talk about de-extinction, we're talking about functional de-extinction. We're identifying the key traits that define those species, and our goal is to create sustainable populations that help communities and ecosystems become more robust and resilient. To do that, we don't need something that's identical to something that used to be alive. We just need something that can solve the problem that is left there because of their extinction. Look at the dire wolves, for example. We got a lot of pushback because we called them dire wolves and they only have 20 edits. They only have 20 edits. But those 20 edits bring back the key traits that were the targets of my team. They're light color and coat, they're larger, and they have these strong muscles. Because we're working on a different genetic background, every edit that we make carries some risk that it's not going to work on that genetic background. I care about de-extinction. I care about fidelity to these ancient phenotypes, but I care more about animal welfare. And I want these animals to be healthy. So my job is to bring back these phenotypes to achieve functional de-extinction with as few edits as possible, since every edit carries risk. So, yes, I still believe that we are not at a place today where we can bring something back that's 100% identical in every way to a particular individual that used to be alive. But I also think that that is not the point. It is not the goal, and it's not even a good idea.
Mhm. So, Biosciences is working on mammoths, dodos, direwolves, and now even moa. How do you decide which species are worth pursuing from both a scientific and ecological perspective?
And the thylacine, that's the Tasmanian tiger.
Mhm.
So we've selected species that fall along some of the major lineages of the animal tree of life. We have a marsupial mammal, the thylacine, a placental mammal, the mammoth, and two birds, dodo and moa. So from a scientific perspective, we've chosen these because they have different challenges that we need to solve. And as we solve those challenges, we can apply what we learn to a pretty big big part of the animal tree of life as far as conservation technologies. We also choose things that are likely to captivate people's attention, that they want to see, that we can get some excitement about. I think one of the things that I like most about Colossal is that it gives people hope. It's an exciting thing. It's an optimistic thing. And I think a lot of people, I mean, I have a 16-year-old and a 13-year-old, and both of them and their friends, I think, and I don't know if this is true for you as well, but there's this weird feeling of, like, what the fuck, right? Like, this is the world that we're inheriting. Why are you doing this to us? Like, everything is bad all the time. The news headlines are always bad. And now there's this company out there that's saying, maybe not. Maybe we can use tools of biotechnology and become genome engineers, and we can manipulate these species to help them to survive. And I think if we had chosen species like, the Christmas Island rat. No offense, Christmas Island rat. You're a great extinct species. I'm sure you did cool things. But way fewer people are going to care about the Christmas Island rat than they do about the mammoth, right? So there's a little bit of excitement and intrigue, and there's a lot of science that goes into selecting who it is. Oh, and of course, sometimes there are just specific groups of people who really want a species to come back. The MOA project, for example, was launched in collaboration with the Ngaitahu Research Centre, which is one of the largest groups of Maori in the South Island of New Zealand who are leading that project because they want to bring the MOA back so that they can have MOA on their land in the South Island of New Zealand.
Very cool. I definitely think Colossal's just an incredible company, very much optimistic, very much cool, sort of futuristic, something you'd want to buy merch for or something like that.
Well, we
Um,
do have a lot of merch. I'm not even wearing a Colossal t-shirt right now, but my kids do go to school wearing their Colossal shirts, and it's kind of that's cool, but your mom being on YouTube is not cool. So they've got like half cool.
yeah, best of some worlds, maybe. Um, how do you address ethical concerns surrounding de-extinction, such as potential ecological impacts or the allocation of resources away from conserving existing endangered species?
Well, I think as far as ecological impacts, we are having an ecological impact on everything with everything that we're doing, whether we mean to or not. And so I think what this is it's a way for us to have a deliberate impact. We know what the impact is going to be. We can do risk assessments and figure out what's going on. I'm not particularly worried that we're going to bring mammoths back and suddenly they're going to take over the world. Elephants have one baby. It's a 22-month pregnancy, and they reach sexual maturity as teenagers. So it's not like that population is going to take off anytime quickly, right? And as far as taking resources away, I just disagree. We are as Colossal, we're funded by an entirely different suite of people than would normally fund conservation work. This is traditional venture capital in some respects. There are some private investors, but these are not people who would otherwise have invested in traditional approaches to biodiversity conservation. Instead, this is new money to generate new approaches that will help biodiversity conservation. And I understand that people think that we should invest all of our money just into buying up habitats. We have been doing that for a long time. Collectively in the world, we've spent billions of dollars in conservation over the last 100 years, and we're winning a few battles against the extinction crisis, but we are still losing the war. I don't think we need more money to do the same thing. I think we need new weapons. And that is what we are building. We're building new tools with different money that we can use to contribute to the protection and preservation of species and habitats. I'm going to have to go. I have got another meeting. Do you have one more question?
Okay, I'll give you just one last question, which we ask all of our interviewees, which is, how do you define success personally and professionally?
Oof, that's too hard.
Mhm,
I think I define success personally as when I get up in the morning. That's my dogs losing their minds. I
mhm.
think I define success personally as when can you hear them?
No,
Can you
it's just
shut up?
silences.
Hey, it's probably like the UPS man or something. Who knows?
Mhm. I can't hear anything right now.
Okay. I think I define success personally as when I get up in the morning and I'm really excited about what I'm doing. Like my dogs are really excited right now about the person coming to my door. And professionally, when other people are paying attention. I'm
Mhm.
really sorry, they're really loud right now.
Oh, all good.
one small box. All right.
Okay,
How do
so
I define success personally? I define success personally, I think, as if I wake up in the morning and I'm still excited about what I'm doing. And even in my current job, there's some mornings where I would rather just go back to bed. But for the most part, I think, I'm having fun, we're making progress, and more importantly, we're developing tools that we can use to stop species from becoming extinct. And professionally, I guess it's very hard for me to separate those two. Maybe I work too hard and need some hobbies, probably. Maybe I should take my dogs for a walk.
okay, thank you so much for your time. It's been great.
Thank you. Let
And
me
also
know how
just
things go. You're going to edit this, I assume. That's why
yeah,
I.
yeah,
Okay.
yeah, I'll edit it, make it look more professional. But
Okay.
thank you so much. It seems like I was just thinking during researching and this interview, it seems like such a incredible, super dream job that um people would love to have more than anything else. Like a kid would say before they get indoctrinated or something like that, but it's going super well and very, very much a cool company, cool idea. So it's been great
Thank
speaking to
you.
you.
All right. Thanks.
Thank you so
Bye-bye.
much.